Good morning. Let's have a class. So we will finish the slides of lecture 2 and then we'll move on to the slide 3 of exam 1. So when you look at these slides, let's do some of the conclusions. This is what we talk about, is stain technology. And we practiced some in the lab, we'll continue to do the practice, and we also talk a lot in the lecture. So stain technology, first of all, we talk about is a simple stain. Simple stain, we will know the morphology and the arrangement. Then we talk about a very important differentiate stain, which is gram stain. And you will get practice today. Talk about the gram stain, and gram positive and gram negative. Then we talk about lots of the special stain. And the special stain, which is including acid-fast stain, endospore stain, and flagella stain, and the capsule stain. So these, we talk about each of these one by one, and the basic principles. And we practice in the lab will be the simple stain we already did, the gram stain we will do today, and the endospore stain we'll do on Thursday. And the others we're not going to do. Besides of that, we also talk about is a negative stain. So negative stain, as an example, is one of the special stain of a capsule stain, because we're staining the background using Indian ink or nigrothane, and then the specimen showed up. So these are the something we want to just give you some outline here, and you can see the slides, what it looks like. So the gram stain, you see the gram positive, gram negative bacteria, red and purple cell, red cell. Acid-fast stain, you see the red cell is acid-fast, that's coming from the carbon fusion. And the blue stain is non-acid-fast, that is coming from the counter stain with methylene blue. So you see the endospore stain. This is using the malachite green with a specific bacteria is Bacillus. Then the capsule stain. This is very possible is Streptococcus pneumoniae. And the last one is a flagella stain. And that special bacteria is Proteus vulgaris. And we will using this strain in the lab session a lot because most of the biochemistry reaction, this bacteria is positive. Okay? Now next, we want to be very quick to go over what is electron microscope. Electron microscope, if we want to do a comparison, we talk about there is a TEM, transmission electron microscope, and scanning electron microscope. So SEM and TEM. And both of these two we're using is an electron as an energy resource. And because it's an electron beam, so the wavelength is much less. So that's why the resolution power value is very low and the resolution capability is strong. And the difference is pretty obvious. First of all is the magnification. The transmission electron microscope somewhere 10 to 30K. And the scanning electron microscope is only about 5 to 6,000, 5 to 6K. And the characteristics of transmission electron microscope, obviously we can see the internal structure. And the scanning electron microscope, we can only see the surface, which is most likely is the 3D images. Now, both of these two electron microscope, if we have to do at WVU, you have to get training first. And the UPD, which means an instructor who is in your lab or who is a project director has to be trained also. And then in the shared facility, which is a building just across here, so you can using electron microscope to do the research. And it's kind of like you need to get a certification first, then you can do. So we're not going to go over detail here. The something I just want to mention, the sample you have to do is a very fast deep freezing. And most of the time, we use liquid nitrogen to do a deep freezing. And then we have to cut them into very thin pieces, so slice into an extremely thin piece. And then we have to coating. At this moment, we will not say it's a stain, we will say it's a coating. Coating usually uses a silver metal, very similar to the flagella stain. Using the silver metal, then we put onto the machine of the electron microscope, put onto the chamber, which is we call it an amplifier, then we can see the structure. So we just very briefly tell you what it looks like. And you can see this is like a cartoon movie picture here. It's a transmission electron microscope. You can see the internal structure. Scanning, you can see the surface, like a 3D images. And if you want to see more detail, this is the ultrastructure of a thin section of TEM. You can see the ribosome there. You can see the cell wall and the nucleates. So this is give you an overall impression what it looks like using TEM. And you also see parasites. If we use SEM, you can see the 3D images. So just give you an overall picture what it is. The last slides we want to talk about is the shape and arrangement. Now, by the way, when we talk about the morphology, usually which means the shape of the bacteria. So we have a terminology, we call it a morphology. This means shape of bacteria. And one of the shape of the bacteria, obviously you already see in the lab, is like a circle. That is coxa. But there is so many of the coxa. So for example, Staphylococcus, Streptococcus agalactiae. This one we will mention about. It's a group B Streptococcus. This is the coccell in chain. And this is Staphylococcus aureus. It's a coccell in the cluster. And this is a grape shape. We do the gram stain today. We already see under microscope, use pre-made slides. And most of the Streptococcus is chain. And the Staphylococcus is grape shape. But sometimes that could be 4 of them stacked together. Sometimes 4 of them will stack together. We call it a tetrad. This is like microbacteria, Ledius. Micrococcus Ledius will be a tetrad. Or some very special bacteria, they even could be either 6 of them stacked together. Okay, that's called succinate or succinate. That's a cubic configuration of 8 coccella. Now, why this is important? Because when we move to the lecture 1, exam 2, we're going to talk about the human pathogen disease, which means the pathological bacteria cause a human disease. They are very typical for their characteristics. Streptococcus ammonium, for example, is diprococcal. Streptococcus pyogenes and agalactiae is a chain. Staphylococcus aureus, epidermis, or saprophyticus is grape shape. So all these things are very typical. Then when you do a clinical identification under the microscope, when you see these bacteria isolated from a patient, you know what it is because you have to use antibiotics immediately. So that's why this gives you overall picture of what it is. Now we talk about the coccell. Then how about this non-coccell stuff? Lods, for example, bacillus, Coccobacillus especially. Coccobacillus here, you will see this. This looks like a bacillus, but it looks like a coccell. Very short lots. So we call it a Coccobacillus. We will mention this lots of time in our exam 2, lecture 3 or 4. For example, Neisseria gonorrhea or Neisseria meningitis is a Coccobacillus. Looks like a coccell, but it is a bacillus. Now vibrio, when you see here is a vibrio chloreal. That's a comma-shaped vibrio. This is usually related to seafoods or lots of the food products in the deep ocean, like a fish. And that will cause a chloreal of the people who is get contaminated or infected. And they are very typical example, like a comma shape. And this is Corynebacteria, this is Capillobacteria jejuni. We will mention this in the exam 4. This is a major pathogen in the chicken products. So chicken products has two major pathogens. Number one is Salmonella, number two is Campylobacter jejuni. And this one we already mentioned. It's one of the example of a spiral sheets is Leptospira interrogans. It's like a hook shape. They will causing like a jaundice of the eye, kind of like a liver disease. And this is a typical example using dark field microscope. And you can see the specimen comes out and the background is showing you like dark color. So all these examples, this is the Bacillus nectarian, kind of like a surrogate bacteria. It's a long chain, like you watch the Bacillus subtilis, it's a long chain longs. So all these examples, you can see the shape and the arrangement is very important because it's very typical to all the pathogens. Plus, we will do the gram stain. Then we know gram positive, gram negative will be a big picture of the morphology shape of the bacteria. And that is usually the number one step to identify a bacteria. The number two is biochemistry reaction. Number three is, of course, is genetics, which means PCR, multiplex PCR, real-time PCR. Then the number four will be the whole genome sequencing, which means you get a single colony, you can do the whole sequencing. The ATGC, the arrangements, the sequence, that will be in an advanced level. Okay, so just want to let you know, shape arrangement is belonging to lots of the characteristics to preliminary identify a bacteria. So that's why it is important. Okay, so we finished here. And I would love to briefly introduce what we have here on the eCampus. So I usually put a lecture slides. Then I will put a Zoom recording, which is I did in 2020 when we completely do online. So if you like it, you can read it. Basically, we go over all the slides. Then I have gonna put in my YouTube channel the link from the classroom teaching. So if you're missing, you can continue to watch. Okay, so that's what I did. All these organizations for the eCampus. Now today we're gonna move on to is chapter 3. Chapter 3 we're gonna talk about the bacteria structure, and this is a very important chapter when we talk about that. So let's talk about these one by one. Okay, first slides is a very basic question. Long time ago, I had this one in the exam, which is what is the major difference between a bacteria and a fungi? Or we can say a major difference between a prokaryotes and the eukaryotes. What's the major difference? If we say it very simple, prokaryote, eukaryote. Okay, say it very simple. What is the major difference? Cell, membrane, bundle, system. Prokaryote doesn't have, eukaryotes have it. So, which means you have a cell membrane system to separate each of the individual cell machinery, and they can work individually and make the life is more prolific. For the bacterial cell, it is more simple because there is no individual cell machinery, in the exception of those DNA stuff, like chromosome, like ribosome, all those genetic stuff. Okay, and there is a theory which can explain why eukaryotes have so many complicated cell machinery there. Because they think a long time ago there is a fungi, or there is a mold or yeast. They are eating a bacteria. Then the bacteria are going to stay there and they integrated the genome sequence into their own genome sequence and later on developed by their own functional machinery. That is a theory called endocytosis. And the same thing for the bacteria. Why bacteria has a toxin? Because the bacteria eat a virus, and the virus integrated their DNA into the DNA chromosome of a bacteria, then become a toxin. That is called a lysogenic conversion. And we will mention this when we talk about the virus in our exam 4. Okay, so just very simple to tell you the difference. We're not going to go over everything for the eukaryote cells because that's what you learn in your plant science class, animal science class, or general biology you already talk about. So we will focus something more specific for a bacterial cell. This is the one which is for prokaryote cell structure. If we look at these details, you will see there is a capsule outside. Okay, then you have a cytoplasma, then you have a ribosome, cell wall, plasma membrane, plasmid, flagella, you have a fimbrial, and you have an inclusion. The inclusion usually is a place you're going to storage some of the chemicals. Let's say in storage phosphate. Okay, so this is the overall picture of a prokaryotic cell structure, what it looks like. And this is another important slide because it gives you an overall picture of the prokaryote cell structure. So you look at the cell membrane. We will talk about all these details. What it looks like could be a barrier, a boundary system, a transporting nutrients take out of the waste, metabolic process location, the detection of the environmental signals. So it looks like it is a sensor. And the cell wall, they tell you a shape and a protection. Ribosome, of course, that's a location for the protein synthesis. And the capsules, they adhere to the surface, so they're going to resistant the phagocytosis. Phyli, which is attachment, they are going through the genetic communication, we call it a conjugation. And they contribute to limited of the capability of the motility. Flagella, major for real movement, we call it a motility, and the inclusions, it's a storage of the granule. So they can be storage, carbon, phosphate, and other of the chemicals. So this is just give you an overall picture, what it looks like. Now we want to talk about each of these one by one. First of all is a bacterial cell envelope, which is encoding a plasma membrane, cell wall, and the layers outside the cell wall. So we want to talk about these one by one. Lots of stuff. We will talk beyond the slides, but to let you know very clearly. Okay, we already mentioned a name for a cell membrane system. A terminology to well describe it called fluid mosaic model. This is everybody knows. In your biology 101, you already learned about that. Then what it looks like, and we mentioned, we can draw the picture again. And something we want to tell you, you have not learned before. It is composed by phospholipids. And the phospholipids, usually they are across the cell membrane system, and they usually will be with a protein. And the major, there is two types of the protein. Some of the protein is right in the middle. So we call it a transmembrane protein. And there are some of the protein could be attached on the side. These type of thing is also important. We will mention real quick. This is called a peripheral protein. Now, what are the major components of these? What are the percentage of that? Transmembrane protein, around 70 to 80% of the whole cell membrane system, and peripheral protein is around 20 to 30%. But it is important. And then you have these lipids. Is it right? This is what the lipids looks like. It's called phospholipids. Phospholipids, it's a polar system. And we had a polar head and a non-polar tail. So this ends up with the polar on the head, which is, let's draw again, right here. Okay, this head, we call it, we call it like water. So we will say it is hydrophilic. Which means they like water. And the tail dislike water, so we call it a hydrophobic. They dislike water. Or we say it is lipophilic. Because they could be dissolve in some of the chemicals, they could dissolve the lipids. Let's say ethanol. Okay, so we all call it lipophilic. Now the question will be, you know about these things. It's a very nice structure because they have a hydrophilic head, hydrophobic tail, and you have a protein there. That's why they can always keep moving. That's why we call it a fluid mosaic model. Now, why this looks like that? That is something we want to tell a little bit detail. Why you have a head is hydrophilic, like water. Why you have a tail is hydrophobic, dislike water. Dislike water, which means it's not going to be easily dissolved in the water. So the story is that we have to look at the chemical structure. So at the top, what it looks like. What is this? Okay, when we talk about that, we need to know about this basic stuff. Can somebody tell me what is this? That's ethanol, is that right? Okay, most of the time we modify it. So, we call it acetylmaramic. When you have this acetylmaramic at the head, kind of like a modified ethanol, the ethanol can dissolve in the water, is that right? Very well. Otherwise, we won't have 95% alcohol and 75% alcohol, and you won't have a grape wine or red wine. So that's why it's dissolved in the water. That's obvious the reason. What's the next structure there? Of course, we need this guy. What is this? We need a phosphate. Is that right? Obviously, otherwise we won't say it's a phospholipids. What we need later? We go here, we're going to have a CH2, CH, CH2. So we go O, O, C. Then we go CH2, CH2. Go to the end, CH2, 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 CH2. What is this structure right here? Glycerol. Is that right? How about here? Go down right here. This could be 15. So CH2. CH2 could be bracket 15. What is this long chain? That is long chain fatty acids. What is that looks like? Think about the oil. Your butter. Is that going to dissolve into water? Of course not. Otherwise you want to have egg mayonnaise. Remember the egg mayonnaise on the top of surface? That's why it doesn't like water. Because it's a very special structure. It goes here. You may not like it because it's organic chemistry. Next time, you're going to get A to take in my class, or you get organic chemistry A first, then take in this class. Is that okay? We're going to put the prerequisite requirements there. So when we talk about it, but when you know this knowledge, you can understand why the head is hydrophilic, the tail is hydrophobic. So the reason is at the beginning it's a modified ethanol. Then become a phosphatic glycerol at the back is a fatty acids. Something like you put the oil there. So that's why it's not going to dissolve in the water. That's why it dislikes water. Okay? So that is a fluid mosaic model. Now the cell membrane, now of course antimicrobials could be disrupted and dissolved by a layer, but be careful. Wheat really doesn't like the antimicrobials can damage the cell membrane system. It is usually an alternative or a backup. Why? Because you are killing bacteria at the same time you are killing animal and human being, because human being has cell membrane. Is that right? So that's why any of the antimicrobials or antibiotics, if they can kill, if their target is cell membrane system, most likely they have a very strong side effect on the human being. Let's say tetracycline. You're going to have a hearing loss. Some of them you have like yellow teeth, because they are attacking cell membrane system. Cell membrane, human being has it. We don't like it. Okay, now the membrane proteins performing aiding in the function, the cell wall synthesis, energy, metabolic. We'll talk about the metabolism in real detail in the examination 3. DNA replication, we will talk about in the examination 3. The censoring and the molecular transport, we will talk about the molecular transport real quick. Okay, now, remember, we talk about the protein right here. Lots of the function for the protein. Could be support, could be a sensor, could be a place for the metabolic reaction. But more important here, this is going to be the uptake of nutrition. Then we're going to move on to the next topic, is uptake of nutrition. And you heard about this guy millions of times, even in your high school. But we would love to say something a little bit different, typically for bacteria. Okay, what are the 3 major things you know about the uptake of nutrition from outside of the cell membrane or cell wall system? Number 1 is very easy, called passive diffusion. Or diffusion. So simple this is. What's the characteristics for that? From high concentration to low concentration. No carrier and no energy currency, which is no ATP. This is so simple, the way to do. What are the chemicals they use in passive diffusion? Water, oxygen, carbon dioxide, so those very basic chemicals. They're using passive diffusion to do. This is the first one, everybody knows. Second one is we call facilitated diffusion. Facilitated diffusion, same thing. It is from high concentration to low concentration. However, they need a carrier. This carrier most of the time is an enzyme. And they do not need ATP. So, if we want to draw a picture of what it looks like, let's say this is a concentration gradient. This will be a transfer rate. What will the curve looks like? Passive diffusion, like this, very straight. This is passive diffusion. What is a facilitated diffusion? Will be like this. It will become flat because this is an enzyme. At the end of the day, the amount of the enzyme will be flat. Okay, this enzyme usually is a carrier. That's a carrier. Lots of the time is a protein, like a bridge across the cell membrane system. Now here is the question. Does the bacteria using facilitated diffusion a lot when they are uptaking the nutrition? Not really. This is very few for bacteria. Why? Because in the real life, the bacteria always survive in a poor nutrition environment, which means they could be surviving on the floor, on the desk. There's not many nutrition you can take besides a residue food soil. We call it a food soil, like you have a little bit sugar, a little bit of carbohydrates, a little bit liquids, lipids there, protein. But majority of the time in the environments, their nutrition is poor. So you're not going to get lots of them outside of the cell membrane system. So this is very few for bacteria. Most of the time for eukaryotes, because they can uptake it. Okay, so facilitated diffusion is very few for the bacteria. For the bacteria, they are using this guy sometimes, which is called active transport. Okay, active transport going opposite direction with the concentration. They go from low concentration to high concentration. This is more realistic for bacteria because the bacteria environment is poor nutrition and they take the nutrition there. And they need energy, which is they need ATP, and they need a carrier. And the majority of the time, the model, we call it ATP binding cassette transport system. And acronym ABC. So we call it ABC transport system. And how they do it, it is a very simple example. We just draw right here. Okay, let's say I have a protein right here. This will be a protein. It's an intramembrane protein. Of course, you have these phosphates there. We just draw a couple as an example. Now, this moment, we need this guy here. These are peripheral protein. Once the nutrition comes out, or sometimes it's outside, okay, it depends, could be inside. When the nutrition comes, let's say they have a star come out, this guy comes in. What's going to happen? These guys will be like an enzyme to let ATP hydrolyze, become ADP, release a phosphates. The same thing right here is ATP hydrolyze becomes ADP and release phosphates. And that's called the ATP binding cassette transport system. And the bacteria using them a lot. However, it is not really a major transport system. In the microorganism area, a major transport system is group translocation. In order to understand this guy, this group translocation system, you need to know the story behind a little bit of the biochemistry. And I believe it's taught you in the Biology 101 or the other classes. So we want to talk about right here. That is called group translocation. Okay, group translocation, what it looks like. They require a system. It's called PEP. What is this? Phosphor enol pyruvate. Once you have this guy, you're going to donate one phosphate. And this phosphate will go to a second one. What is that? It's a complex called E1. That E, which means enzyme, I, which is a complex one. So this is, you can say, this is the enzyme complex 1. And not enough, they're going to go here. This phosphate's going to go here. This will be H or P. What is this guy? Heat stable protein. And at this moment, when they transfer those 3 steps, this phosphate will be released. And what they do, they go through this cell wall system right here. Okay, now we're going to have to do like this. That's why I tell you, you have to dry all these things. We dry a couple, okay, not everything. There is a lot of the time, there is a difference in these boxes right here. This is called, all these things we call, it's an enzyme 2 complex. And these enzyme 2 complexes will be helped if there is a sugar, we call it a glucose, comes out from the cell outside of the cell membrane, go inside. Then they will directly become glucose 6-phosphates. In your slides, if there is a mannitol, they come in, will directly become mannitol 1-phosphates. Now, by the way, mannitol is a special sugar. We're going to talk about that later on. Let's say mannitol salt agar is a specific bacterial agar for Staphylococcus aureus. Now the question is, why they have to do that? This is need to know a basic biochemistry cycle. Do you still remember what is this guy comes from? Where you see these? Where you see these? There is a very glycolysis, if you remember. What is glycolysis? Glucose will become glucose 6-phosphate. Then become an isomer, fructose 6-phosphate. Then become fructose 1,6-biphosphates. Is that right? Then this stage we go to a branch. What are they? Glyceraldehyde 3-phosphates. Then we go 1,3-biphosphoglycerate. Then go 3-phosphoglycerate. Then go 2-phosphoglycerate. And then the last one is phosphoenolpyruvate. And later on it becomes a pyruvate. And this pyruvate goes through the circle called TCA cycle. Look at here, glucose 6-phosphates. And look at here, phosphoenolpyruvate. Why they can do this? Bacteria is a very short lifetime, is that right? 24 hours. Microbacterial tuberculosis may be longer, about a week. They're not going to stay there forever. You will talk about the growth curve real quick. 24 hours, 48 hours, they're going to die. In their very short lifetime, they want to use the energy is more efficiency. So they have to go through this metabolic cycle more quickly. Therefore, if your glucose comes in, I want you the glucose go in, is directly become glucose 6-phosphates. Then you can go through these processes more efficient compared to only a glucose going. And then at the end of the day, at the end of the glycolysis, you have a phosphoenolpyruvate. This phosphoenolpyruvate, that phosphates is loosely attached. Then they can be released and do the more contribution. So this is a cycle like. This is actually like a cycle. Here is group translocation. Because when they do that, the bacteria could use the energy is more frequently. So I'm going to tell you, for the bacteria takes the nutrition, the more efficient way, the more frequency what they use is group translocation. It's not those facilitated diffusion, like active transport. They may be used a lot, maybe used a little bit, not as much as we talk about the group translocation. So you always remember about this. And this is also a connection between when we talk about the exam 1 and exam 3 when we talk about biochemistry information. Okay? So here, if you look at the slides, this system called Phosphor-transferase system. When you see ASE, which means it's an enzyme, and that magic chemical is phosphoenolpyruvate. They will donate the phosphate to going through multiple complex. And this complex looks like a group. So that's why we call it a group translocation. And they will let glucose mannitol directly become glucose 6-phosphate or mannitol 1-phosphates. They will jump in, glycolysis is more efficiency and more quickly. Now you're going to ask me the mannitol 1-phosphates. Mannitol 1-phosphates have a similar glycolysis. It's called a modified glycolysis. And you can see some textbook, when we talk about microbial physiology, they will talk in detail about that. Okay, that's group translocation. Next, we're going to have to tear these off. We're going to talk about the bacterial cell wall. We already mentioned some of that when we talk about the gram stain, both in the lab and in the lecture. And we will go detail some of them about today, talk about the major components of this bacterial cell wall. So first of all, this is our conclusion picture. It's important. This is also tells you a cell wall theory. Now, why bacteria is going to stain positive? We call it a gram positive. Why stains purple? Because they have a thick peptidoglycan. And why gram-negative bacteria stays pink and red? Because they are very thin peptidoglycan. And the peptidoglycan is a rigid structure outside of the cell plasma membrane. Okay, so overall, let's talk about what's the cell wall function. Number one, maintaining the shape. Every bacteria need to have the shape. We talk about the Koch cell rods. The maintaining of the shape is rely on the cell wall. They will be help protect the bacteria from osmotic lysis. What means osmotic lysis? If you put a bacteria into a distilled water, it will be bursted. So let's do an example. If you put 1,000 bacterial cells in a distilled water, after enumeration, you will find it's maybe only 500. So where all the other 500 go? They're dead. So the reason is the unbalance of the pressure between the cell wall and the cell membrane system, compared outside and inside of the cell. So they burst. So that's called osmotic lysis. That's a cell wall function can protect that. Now, of course, it protects some of the toxication material, let them more difficult to come in, and they may contribute to the pathogenicity. This time we talk about is Lps Lipopolysaccharides, which is in the gram-negative bacteria. And remember, I draw the picture of the gram-negative bacteria right there when we talk about the gram stain. This is the first picture, and it's an important information when we talk about the peptidoglycan. Okay, what is peptidoglycan? We talk about it's a rigid system just outside of the cell membrane. So you have these slides look like here, it's so complicated. This curve, that curve, hard to understand sometimes. We can write a very simple sentence to describe this, and then we'll talk about the detail. What is the major chemical components of peptidoglycan? It is composed by modified sugar, cross-linked with amino acid. That cross-link could be a simple chain, or it could be a bridge. Or a bridge. Okay, the next question. What are those modified sugars? There are two major modified sugars. Number one, NAM. What is that? Acetylmaramic acids. What is that looks like? Okay, then we go chemistry again. What it looks like? At the beginning, you need to know this guy. What is this? OH, H, H, OH. Opposite direction, OH, H, go, OH, H, then go, CH2, OH, and then H. What is this? Glucose. If you don't know that structure, retake you back organic chemistry. Is that okay? It doesn't make sense if you get A, you don't know that glucose. Is that okay? I make joke to you all the time because the people tell me, unless your nickname name is Albert Einstein, you always get F for D in your organic chemistry. Is that right? But we have to know something, a basic knowledge. So it's a glucose. So what is N-acetylmaramic acids? There is two things going to be happened. This is carbon number 2. This guy is gone. This guy with NH2, NHCOCH3. Okay. This is a acetyl. How about here? Take this O goes here. Is here we'll have a CCH3H and then go COOH. This is N-acetylmaramic acids. Okay, what are the other one? Is NAG. What is that? That is called N-acetylglucosiamine. What is that? This is so simple. That's not difficult at all. This guy right here. OH, H. Right here. Same thing. Take this guy out, NH. N-acetylglucosiamine. How they connected with each other? Carbon number 1, carbon number 4. Go. 1,4 glycosidic bonds. They connected. Oh, of course, there's got to be a long chain. Okay, one by one from here. Very, very long. But basic structure is N-acetylmaramic acids and N-acetylglucosiamines connected with each other with 1,4 glycosidic bonds. Make sure this is going to have a turn. Okay, I just draw it simple, like this. Okay. Okay, when you have this guy, what's going to be there? Then where are going to be the amino acids go? So usually we're adding the top. We have MAM, we have NAG. Then we're going to connect it with something. People do a very special research in gram-positive bacteria. They find that the majority of them, they will connect it with L-alanine. And then they will go with D-alanine. Oh, sorry. They will go with D-alanine.